What if I told you the real reason colon cancer is rising so aggressively in adults under 50 has less to do with bad luck and far more to do with what happens inside the colon every single day? Not genetics, not fate, not some invisible curse hidden in your DNA, but the environment your colon cells are forced to live in. And here's the terrifying part. Most people are unknowingly feeding the exact biological conditions cancer cells need to survive. Right now, as you're watching this, there are two different battles happening inside your body. One side is trying to protect you, the other side is quietly creating the conditions that allow disease to grow. The foods you eat determine which side wins. For decades, people believed colon cancer was mostly hereditary, something that simply runs in families. But modern research is painting a very different picture. Scientists now estimate that the majority of colon cancer risk is linked to lifestyle and dietary patterns, especially the long-term health of the gut microbiome. That changes everything. Because if the environment inside the colon can help cancer grow, it can also help stop it. And that's where this becomes fascinating. After nearly 3 decades studying cancer biology and metabolic disease, one fact continues to shock people the first time they hear it. Cancer cells are weak. Not weak in appearance, not weak in danger, but weak metabolically. Unlike healthy cells, colon cancer cells become incredibly dependent on one specific fuel source: glucose. They burn sugar aggressively and inefficiently to survive. At the same time, they rely heavily on inflammation and rapid blood vessel growth to expand. Healthy colon cells behave very differently. They're flexible, adaptable, resilient. They can use multiple fuel sources for energy, including one remarkable compound most people have never heard of, a short-chain fatty acid produced inside the gut during fiber fermentation. And this is where the story takes an extraordinary turn. Because the very compound that strengthens healthy colon cells has been shown to stress, weaken, and even trigger self-destruction pathways in abnormal colon cells. Same environment, same colon. 2 completely different outcomes. This is why some researchers now describe the colon as an ecosystem more than an organ. What thrives there depends entirely on what you repeatedly feed. A few years ago, I met a woman I'll call Margaret. She was in her early 60s, active, healthy weight, walked daily, didn't smoke, rarely drank alcohol. By outward appearance, she looked like someone doing everything right. Then, a routine screening changed everything. Doctors discovered multiple polyps during her colonoscopy. Most were harmless, but one showed early cellular changes associated with precancerous development. Naturally, she panicked. Most people would. She immediately asked the same question nearly every patient asks. How did this happen if I eat healthy? But when we looked deeper, the answer became obvious. Her gut microbiome, the bacterial ecosystem living inside her colon, was severely depleted in beneficial bacteria responsible for producing protective fermentation compounds. In other words, her colon looked healthy from the outside, but internally, the protective systems designed to defend her colon had been starving for years. That distinction matters more than people realize. Because colon cancer doesn't usually appear overnight. It develops slowly, in an inflammatory environment that gradually weakens the colon's natural defense systems. And modern diets are accelerating that process. Highly processed foods, refined sugars, excess seed oils, low fiber intake, chronic stress, poor sleep, sedentary lifestyles. All of these factors shift the gut microbiome in ways that promote inflammation while reducing the production of protective compounds. The colon becomes chemically hostile to healthy cells and increasingly favorable to abnormal ones. But here's the hopeful part. The opposite is also true. When you begin feeding the right bacteria, the chemistry of the colon starts changing surprisingly fast. Inflammation begins decreasing. Protective compounds rise. Immune signaling improves. Even the ability of abnormal cells to survive starts weakening. And one of the most important compounds involved in this process is something almost nobody talks about. It's called butyrate. This single fermentation compound may be one of the most powerful natural colon protective substances ever discovered. Healthy colon cells absolutely love it. Cancer cells struggle to survive around it. But here's the catch. You don't directly eat butyrate itself. Your gut bacteria have to manufacture it for you. And whether they can do that depends entirely on what you consistently put on your plate. Most people think cancer cells are unstoppable. Like once they appear, they simply grow stronger and stronger until nothing can slow them down. But that's not what the science actually shows. Cancer cells are powerful in some ways, yet shockingly vulnerable in others. And one of the biggest weaknesses colon cancer cells have is something researchers have been quietly studying for years. They are metabolically trapped. Healthy colon cells are flexible. They can switch between different fuel sources depending on what the body needs. They can burn fats, amino acids, glucose, and most importantly, a fermentation compound produced by beneficial gut bacteria. That compound is called butyrate, and healthy colon tissue depends on it more than most people realize. But cancer cells, they lose that flexibility. Instead of efficiently producing energy like healthy cells, they become addicted to rapid sugar fermentation through a process scientists often call the Warburg effect. Even when oxygen is available, these cells continue burning glucose in a chaotic, wasteful way because it allows them to grow quickly. But that addiction creates a dangerous weakness. Because while healthy colon cells thrive in a butyrate-rich environment, abnormal colon cells often cannot process it correctly. And what happens next is one of the most fascinating discoveries in modern cancer biology. When butyrate levels rise inside the colon, healthy cells absorb it as premium fuel. It strengthens the lining of the colon, supports repair mechanisms, and reduces inflammation. But inside abnormal cells, the same compound behaves completely differently. Instead of fueling the cell, it begins interfering with the genetic machinery that allows the cancer cell to survive. Imagine that for a moment. The exact same molecule feeds healthy tissue while stressing damaged tissue. That level of biological selectivity is extraordinary. Researchers discovered that when certain colon cancer cells are exposed to high concentrations of butyrate, they begin activating self-destruction pathways. In simple terms, the cancer cell starts initiating its own shutdown sequence. Not because of chemotherapy, not because of radiation, but because the environment around it changed. This is why the gut microbiome matters so much more than people think. Because your colon is not just digesting food. It's constantly manufacturing chemical signals that either support protection or support disease. And most people are unknowingly starving the very bacteria responsible for producing these protective compounds. Here's where it becomes even more important. Butyrate is not found sitting on grocery store shelves. You cannot simply buy it in a bottle and expect miracles. Your body must produce it internally through bacterial fermentation. That means your daily food choices determine whether these protective bacteria survive or disappear. Certain fibers and resistant starches reach the colon undigested. Once there, beneficial bacteria ferment them and produce butyrate as a byproduct. But if those fibers are missing from the diet long enough, the bacteria themselves begin disappearing. And that's exactly what researchers are now seeing across much of the modern population. Lower microbial diversity, lower butyrate production, higher inflammation, higher rates of metabolic disease, and increasingly, higher rates of colon cancer in younger adults. This explains why two people can eat what looks like a similar diet, yet have completely different internal environments. One person's gut ecosystem produces anti-inflammatory compounds daily. The other person's ecosystem produces inflammatory chemicals that damage the colon lining over time. Same calories. Completely different biological outcome. A patient once told me something I'll never forget. She said, "I thought my stomach only processed food. I never realized it was also controlling my future." That statement captures the entire problem. Most people think about food only in terms of weight gain or weight loss. Very few think about the invisible chemical environment being created inside the colon every single day. But cancer cells absolutely care about that environment. And there's another critical detail most people never hear. Tumors cannot grow beyond a tiny size without building their own blood supply. This is one of the most important concepts in cancer biology. A microscopic cluster of abnormal cells can remain harmless for years unless it successfully recruits blood vessels to feed it. Once that blood supply develops, growth accelerates dramatically, which means one of the body's greatest anti-cancer defenses is the ability to block unnecessary blood vessel formation before tumors expand. This process is known as anti-angiogenesis. And astonishingly, some of the most powerful, natural anti-angiogenic compounds ever identified are found in everyday foods. Certain plant compounds help create an environment that makes it harder for abnormal tissue to establish new blood vessel networks. In other words, some foods don't just support healthy cells. They actively interfere with the biological systems tumors depend on. And once you understand how this works, you stop looking at food as entertainment. You start seeing it as information, signals, instructions, chemical messages that either strengthen protection, or strengthen disease. Here's something most people never hear from their doctor. Your body already has built-in anti-cancer systems operating inside you right now. The problem is not that these systems don't exist. The problem is that modern lifestyles slowly weaken them. And once these protective systems begin breaking down, abnormal colon cells gain the perfect environment to grow, spread, and eventually become dangerous. But when these systems are properly supported, something incredible happens. The body becomes dramatically harder terrain for cancer to survive in. Researchers now focus heavily on three major biological defense systems connected to colon protection: butyrate production, anti-angiogenesis, and NRF2 activation. These three systems work together like a coordinated defense network. When all three are functioning properly, inflammation decreases, cellular repair improves, and abnormal growth becomes much more difficult to sustain. Let's start with the first system, butyrate production. As you learned earlier, butyrate is a short-chain fatty acid produced when beneficial gut bacteria ferment resistant starches and certain fibers inside the colon. Healthy colon cells use butyrate as fuel, but abnormal colon cells often struggle in its presence. And this isn't theoretical anymore. Multiple laboratory studies have shown that higher butyrate concentrations can influence genetic pathways involved in cell cycle regulation and programmed cell death. In simple terms, the environment inside the colon begins favoring healthy tissue instead of abnormal tissue. But there's another layer most people never hear about. Around 70% of the immune system is connected directly to the gut. That means the health of your microbiome influences far more than digestion. It influences inflammation signaling, immune surveillance, and even the body's ability to recognize abnormal cells early. When beneficial bacteria decline, immune communication weakens, inflammation rises, and protective signaling begins collapsing quietly in the background. That brings us to the second system, anti-angiogenesis. This may be one of the most important cancer prevention mechanisms in the human body. Because tumors cannot become dangerous without blood vessels, a tiny cluster of abnormal cells is limited until it recruits circulation to feed its growth. Once that vascular network develops, expansion accelerates rapidly. So, the body constantly works to regulate unnecessary blood vessel formation. And certain natural compounds appear remarkably effective at supporting this protective balance. Green tea is one of the most studied examples. Its active compounds, called catechins, have been extensively researched for their role in regulating inflammatory signaling and vascular growth pathways. But preparation matters more than most people realize. Boiling temperatures can damage delicate compounds inside green tea, reducing its biological activity significantly. Gentle brewing temperatures preserve far more of the active protective compounds researchers study. And then there's garlic. Most people use it only for flavor. But crushed garlic produces a sulfur compound called allicin, which researchers have associated with anti-inflammatory and cellular protective effects. Here's the catch nobody talks about. Garlic must rest after crushing. When garlic is chopped or crushed, enzymes activate chemical reactions that create allicin. But if heat is applied immediately, much of that process never fully occurs. That means preparation changes potency dramatically. Now let's talk about the third system, NRF2 activation. This system is often described as the master switch of cellular defense. When NRF2 activates properly, the body increases production of detoxification enzymes, antioxidant defenses, and DNA repair pathways. In other words, it helps cells defend themselves against damage before problems escalate. And one of the strongest natural NRF2 activators researchers have identified comes from cruciferous vegetables, especially broccoli sprouts. Not mature broccoli, broccoli sprouts. These tiny sprouts contain extremely concentrated levels of sulforaphane, a compound studied extensively for its ability to activate cellular protection systems. What shocked researchers was how powerful the effect appeared even from food quantities achievable in everyday life. Certain studies observed measurable biological changes after participants consumed broccoli sprouts consistently for several weeks. Not supplements, not pharmaceuticals. Food. But again, preparation matters enormously. Heat destroys the enzyme needed to activate sulforaphane efficiently. That means heavily boiling broccoli or sprouts may eliminate much of the protective activity people are hoping to get. This is why some individuals say, "I've eaten vegetables my whole life," yet never experienced the biological benefits researchers describe. The food itself wasn't necessarily the problem. Preparation was. And this is the pattern you keep seeing again and again in nutritional science. Tiny details create massive biological differences. Now here's what truly stunned me after years studying this field. These systems don't work independently. They overlap. The same foods supporting butyrate production also influence inflammation signaling. The same compounds helping activate NRF2 also support anti-angiogenic activity. The microbiome itself influences immune communication tied directly to tumor surveillance. It's one connected ecosystem, which means every meal either strengthens that network or weakens it. And unfortunately, most modern diets weaken it daily. Processed foods, refined carbohydrates, low-fiber eating patterns, excess sugar, highly inflammatory oils. All of these contribute to an internal environment that gradually favors disease progression over protection. But the encouraging part is this. The body begins adapting surprisingly fast once the environment changes. Microbial populations shift. Inflammatory markers improve. Protective compounds increase. And the colon begins rebuilding a healthier chemical environment from the inside out. Now that you understand the three major systems involved in colon protection, the next question becomes obvious. What exactly should you eat to support them? Not vague advice. Not eat healthier. Not generic nutrition slogans. Specific foods. Specific preparation methods. Specific biological effects. Because once researchers started studying long-term colon health more closely, a fascinating pattern emerged. Certain foods repeatedly appeared in populations with lower inflammatory markers, healthier microbiomes, and reduced colorectal disease risk. And when scientists examined these foods inside laboratories, many of them showed measurable effects on inflammation pathways, gut bacteria, blood vessel signaling, and cellular protection systems. This is where the science becomes practical. Let's start with the single most important food in this entire discussion: broccoli sprouts. Tiny, simple, easy to overlook. But biologically, they are incredibly powerful. Broccoli sprouts contain concentrated amounts of sulforaphane, one of the strongest natural activators of the NRF2 defense pathway researchers have identified. And here's what makes this remarkable. Just a small serving of sprouts can contain dramatically higher sulforaphane potential than mature broccoli. But preparation is everything. If high heat destroys the activating enzyme, the biological effect drops sharply. That means raw sprouts, or sprouts added after cooking, preserve far more protective activity than heavily cooked versions. Many people unknowingly destroy the exact compound they're trying to benefit from. Next comes resistant starch. This may be one of the most misunderstood areas of nutrition. Most starches break down quickly into glucose during digestion. Resistant starch behaves differently. It bypasses digestion in the small intestine and reaches the colon mostly intact, where beneficial bacteria ferment it into butyrate. Remember what you learned earlier. Healthy colon cells thrive on butyrate. Certain abnormal cells struggle in its presence. One of the easiest resistant starch sources is cooled, cooked potatoes. And yes, the cooling matters. When potatoes cool after cooking, part of the starch structure changes into a form. Bacteria can ferment more effectively. Green bananas work similarly. Not ripe yellow bananas. Green bananas. Because the resistant starch content decreases as bananas ripen. Then there's turmeric. For years, people treated turmeric as little more than a trendy spice. But researchers became increasingly interested in curcumin, one of turmeric's primary active compounds, because of its relationship with inflammation signaling. Here's the problem most people never hear. Curcumin absorbs poorly on its own. That's where black pepper becomes critical. Black pepper contains piperine, which dramatically improves curcumin absorption. And when turmeric is combined with healthy fats like olive oil, absorption improves even further. Tiny combinations. Huge biological differences. Now let's talk about pomegranate. Most people think of it as just another fruit juice. But pomegranate contains unique polyphenols that beneficial gut bacteria convert into compounds associated with cellular protection and gut barrier support. Even more interesting, researchers observed that certain bacterial populations linked to healthier colon environments appeared to thrive when exposed to these compounds. Timing may matter too. Since gut bacteria remain metabolically active overnight, consuming pomegranate in the evening may support fermentation activity during critical repair hours. Then come deeply pigmented purple and black foods. Blueberries, purple potatoes, black beans, dark berries, These foods contain anthocyanins, plant pigments, studied for their antioxidant and anti-inflammatory properties. What caught scientists' attention was their apparent selectivity in laboratory studies involving abnormal colon cells. Some compounds appeared capable of stressing unhealthy cells while leaving healthy neighboring cells largely unaffected. Again, the environment matters. Next is green tea. Green tea contains catechins, especially EGCG, compounds studied extensively for their role in inflammation regulation and cellular stress responses. But one mistake ruins much of its potential. Boiling water? Excess heat damages delicate catechins. Gentler brewing temperatures preserve more of the compounds researchers actually study. And adding dairy milk may reduce absorption further because proteins can bind to the active compounds. Small details. Massive difference. Finally, garlic. Garlic may be one of the most biologically underestimated foods in the modern diet. When crushed, garlic activates enzymes that produce sulfur-containing compounds linked to anti-inflammatory and protective cellular activity. But again, preparation changes everything. If garlic is heated immediately after chopping, much of this reaction never fully develops. Allowing crushed garlic to rest before cooking dramatically increases formation of these compounds. This is the pattern you should notice throughout all of this. The body responds not only to what you eat, but to how those foods are prepared, combined, and consumed consistently over time. And consistency matters far more than perfection. Many people fail because they approach nutrition emotionally instead of biologically. They expect dramatic changes in a few days. But microbiome shifts take time. Inflammatory pathways take time. Cellular environments take time. The colon lining itself constantly renews and rebuilds based on repeated exposure, which means every meal is either reinforcing protection or reinforcing vulnerability. A single salad won't save anyone. A single unhealthy meal won't destroy anyone either. But repeated patterns create biological momentum. That's what matters. And there's another important point people rarely hear. A healthy colon environment isn't built by adding protective foods while continuing a highly inflammatory diet unchanged. You cannot overwhelm constant inflammation with occasional superfoods. Processed meats, excess sugar, ultra-refined foods, chronic overeating. These factors continuously feed inflammatory signaling inside the colon. Protective foods work best when the overall environment shifts with them. That's the real goal. Not dieting, not restriction. Environmental change inside the body itself. Here's the uncomfortable truth most people never hear. Many people are eating healthy foods while accidentally destroying the exact compounds that make those foods protective in the first place. Boiling broccoli too long destroys much of the sulforaphane supporting activity. Heating garlic immediately after chopping prevents the formation of important sulfur compounds. Drinking green tea with boiling water can damage delicate catechins researchers study for cellular protection. Tiny mistakes, massive biological consequences. But the biggest mistake of all? Quitting too early. Most people expect to feel dramatically different within days, but colon protection works on a cellular timeline, not an emotional one. Your microbiome needs time to shift. Beneficial bacteria need time to grow. Inflammatory signaling needs time to calm down. Real biological change often happens silently before you ever feel it. And there's one more mistake that cancels everything out. Trying to add healthy foods without removing the inflammatory habits feeding the problem. You cannot build a healthy colon environment on top of constant processed foods, excess sugar, poor sleep, and chronic inflammation. The body responds to patterns, not isolated actions. And that means every consistent choice matters more than most people realize. If this changed how you think about colon health, subscribe now, because in the final part, I'm going to bring everything together into one simple framework you can actually use for the next 12 weeks of your life.